This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 12, recorded on July 22nd, 2011. Hi, everybody. I'm Vincent Racaniello, and this is TWiM, the podcast that explores unseen life on Earth. Joining me today from the Medical University of South Carolina, Michael Schmidt.
Hello, Vincent. How are you this afternoon?
Very warm.
Very warm. Welcome to my world as well.
I know you're not very impressed, but it's 104 degrees here in New York City today.
Oh, that is miserable.
It's not usually that hot here. And Con Edison, our power provider, has reduced the power at noon. So we're on emergency power here, actually, at Columbia. It's warm, but it's okay. I like hot weather. I came from Minneapolis, where it was also 104. There seems to be a heat wave coming across the country. Also joining me today from Small Things Considered, I almost said All Things Considered, Small Things Considered in San Diego, Elio Schaechter.
Hi, and I think this place has the distinction of being the coolest city in the United States right now.
72 degrees, right?
Yep, I can't complain.
Does it ever get really hot there?
Oh, occasionally for a couple of days, about 90 maybe.
Yeah, we don't often have 100 temperature.
No.
Usually 90s in July maybe, but this is unusual. It will go away, but it makes things very dry, and of course, when that happens, West Nile is transmitted. But that's a story for another podcast.
Better that than dengue.
Absolutely. That's one thing we don't have. Not yet, anyway. Margaret McFall-Ngai was supposed to join us today. She's having Skype troubles. Maybe she will join us in a few moments. But we'll just get started here. We have two papers which use unusual methods, I think, to achieve two different aims. And I believe we will start with the first one, which was sent to me by Stan Malloy, who said this might be an interesting paper to look at. And it is published in Proceedings of the National Academy of Sciences. The title is Dissection of the Burkholderia Intracellular Life Cycle Using a Photothermal Nanoblade.
No less.
Oh, that's something.
The title by itself is cool. Photothermal Nanoblade.
We'll find out what that is.
Had any of you before known what a photothermal nanoblade is?
No,
it sounded something out of Star Wars.
It's very cool. And I didn't know much about Burkholderia either. So, Elio, can you tell us something about this? Hold on.
Hi, it's Margaret.
Welcome, Margaret McFall-Ngai, to episode number 12.
Thank you.
You sound great. I hope everything is stable.
I hope so, too.
We're just starting to talk about the first paper on Burkholderia using a photothermal nanoblade. Had you ever heard of a photothermal nanoblade before?
Me? I have to admit I had never heard of a
none of us had, so you're in good company. Okay, Elio, take it away.
Okay, well, I'd like to start out by talking a little bit about intracellular life. Not a subject that we've covered yet, but a terribly important one because it includes what many very important pathogens do. TB, Salmonella, Shigella, Listeria, many do that. They live inside of cells in our body. So let's first of all consider, where did it start? Well, I guess that when prokaryotic cells stopped having the world to themselves and eukaryotic cells arose, they were in the form of what we glibly call protists now, unicellular eukaryotes, and many of them obviously live by eating prokaryotes. And this has gone on for about 1.5 billion years. So these are old habits. And so bacteria, archaea, learned in part to survive that. And this has been carried through, and it's the reason why today they survive not free living protists, but the amoebas inside of us, like the white cells of the blood. So many bacteria get eaten by polymorphonuclears and macrophages, and they get killed, but many do not. So the intracellular form of life is terribly interesting, to me at least. Anyhow, so what's it all about? First of all, why do bacteria actually thrive and grow inside of cells? Well, one of the reasons is that they are sheltered. They're sheltered from antibodies, they're sheltered from antibiotics in some cases, they're sheltered from other cells that would eat them. So the intracellular site is a cozy place for bacteria.
Is this, for these bacteria, Elio, that multiply inside cells, do they also multiply outside of cells?
Yes, except for some, right.
They can do both.
They can do both, except for some which have lost a lot of the genes, like Rickettsia, chlamydia. By the way, I don't know if you knew, Coxiella was always counted as something like a Rickettsia, and therefore a strict intercellular parasite. Did you know a couple of years ago people could start to grow Coxiella on agar, make colonies? Fabulous. Anyhow, so yes, there are, the bacteria I mentioned earlier are all capable of making colonies on agar, therefore living outside of cells, but a few are strict intracellular parasite. All right, so one of the problems that bacteria have, which grow intracellularly, is how to spread to other cells. I mean, this may be a cozy place living inside of a cell, but there's not much future in just being inside a single cell. So spreading to others is a requirement for their development. So how do they do it? Well, many do it just like some viruses do it. They lyse the host cell, and they come out and spread to other cells. And that's, of course, one mechanism. But that mechanism exposes the bacteria to, as I said, antibodies and antibiotics and other cells and so forth. So it's a dangerous trip. A more, what should you call it, more careful way of doing it is to go from cell to cell without being transported into the extracellular milieu. And there are many bacteria that do this. And the story here, I'd like to start a little bit by recollecting the story of what bacteria like Listeria do. Well, imagine a Listeria inside of a human cell. Not only will it divide in the cytoplasm, it gets there, by the way, by a complicated mechanism. It's first taken up by a phagosome vesicle, and then it comes out. It goes through the vesicle into the cytoplasm of the host cell. And there, of all things, it moves around. And it moves at very high speeds, 20 microns per second or something like that. And it looks like sort of a random motion going all over the place. But the reason for this motion is not flagellar motility, but rather the polymerization of actin filaments at one end of the cell. One pole of the cell has a protein that binds to actin, and actin deposits there and pushes the bacteria along. It's fun to watch.
Is there a purpose for this movement?
Okay, the purpose for this movement, best we can tell, is that occasionally the bacteria find themselves at the end of a filament made by the protrusion of the cell membrane of the host cell, and this enters into the next cell. It's like it penetrates into the next cell. Now the cell, the bacterium, which is at the tip of this filament, has to go through its own membrane, or the membrane of the cell that it was in, and then the membrane of the next cell. But it does that. And so it spreads into the next cell. And you keep this going, and you have a spread from one cell to another without the bacteria ever seeing the outside.
It's sort of like the buildings in downtown Minneapolis, Vincent. They make these catwalks for people to go through between buildings to avoid the snow. And you got to realize that it takes a long time for humans to make catwalks, but bacteria do this in real time. And it's principally, as Elio said earlier, it's to hide from those evil antibodies and, let's not forget, complement and all those other things that we got running around in our systems. And it really helps them avoid our immune system until such time that they have a critical mass of cells in which they can then cause disease.
But that's a side effect, because the original driving force was these bacteria were being eaten, and then the survivors evolved a way to survive.
Pathogens.
Right?
Yeah.
That's how selection works.
That was before immune systems. But then later on, when they got into organisms with immune systems, this was a benefit that they hadn't realized, right?
Well, one thing that's really interesting to me here, as somebody who sort of thinks in an evolutionary context, is the issue that in most invertebrate phyla that have been looked at, they have members that have beneficial intracellular symbionts. So all of those hydrothermal vent animals and most of the insect symbionts are intracellular and they're beneficial. And also rhizobium is intracellular in root nodules of plants. So one of the things I find really interesting is that it seems like in the vertebrates, the fishes on up, to us and mammals and so on, it's extremely rare for there to be intracellular beneficial symbionts. They're almost always pathogens. And that's
unless you count mitochondria.
Unless you count mitochondria. But I think that's a really interesting dichotomy.
So the symbiosis that Nicole Dubilier studies, for example, in these vent worms, these are intracellular bacteria.
They are.
Whereas in your symbiosis, it's not, right?
That's right. They're extracellular.
Okay.
So there is a point in, so the intercellular life is complex. And your point is great, Margaret, that this is not, it can be beneficial or deleterious, and that there seems to be an evolutionary scale to this, which is not well understood, I guess. I don't know. I don't think I know why there aren't beneficial symbionts of metazoans.
Actually, there are of metazoans, just not...
I meant vertebrates.
Vertebrates. And I think one of the things that's fascinating is that it correlates with the presence or absence of the adaptive immune system, which might be key. But I think... Elio, I think it's just a fascinating thing. Why? I think I've stood up in front of many physician audiences and asked them if they've ever seen a beneficial intracellular symbiont in the vertebrates, and it doesn't seem to occur.
So it's in humans, at least, when this happens, it's a problem. It's pathogenic.
Yeah, that most of the time,
But in animals,
although they're long range associations. I mean, TB can survive inside of macrophages for years and years and years without causing disease. So there is sometimes there's a standstill and a truce is declared.
Well, I think that's a really good point, Elio, because the issue of carriage, intracellular carriage, might be important for tuning the immune system. So things like Toxoplasma gondii as well as M. tuberculosis. What is it? M. tuberculosis or tuberculome?
Yeah, M. tuberculosis,
Yep.
Mycobacterium tuberculosis.
Right.
So anyhow, let's go back to Burkholderia. So the point of this paper is that this is different. This guy lives inside of cells. I'll say something about Burkholderia in a second. But it transfers from cell to cell by a totally different mechanism. And this is what's news. Okay, so first of all, Burkholderia is a gram-negative bacterium. It used to be called a Pseudomonas. It lives in the soil. It's like a good Pseudomonas has immense metabolic capacities. One of them, Burkholderia cepacia, can oxidize hundreds, if not thousands, of different compounds. And it's somewhat been thought of as being useful in things like bioremediation of pesticides and things like that. Anyhow, there are pathogens in here. The main one, I think, is called, of all things, Burkholderia. By the way, Burkholderia is named after a guy named Burkhold, who was a plant pathologist at Cornell. So the granddaddy of them is sort of Pseudomonas, Burkholderia pseudomallei, which causes a disease in people called Maleoidosis. It's a acute disease that can get chronic and it can be lethal. It exists in tropical areas especially. There is also a bug with a reduced genome from B. pseudomallei, which is called B. mallei. And that causes a disease called glanders in horses. Okay, these guys are dangerous enough to be classified as dangerous possible terrorist agent. So they're classified as, you have to work on them under very strict conditions. BL3 is the name of the condition that you have to use. But there is a cousin of them called Burkholderia thailandensis, after Thailand, I guess, which is not so virulent, and it's possible to work on it under BL2 conditions, which is essentially working on the bench with a lot of precautions.
Hey, Elio, do you know what Maleoidosis is derived from?
I'm sorry, I should have looked it up. I love derivations. Do you happen to know?
Yeah, I have it here. It's from the Greek malus, which means a distemper of asses.
A distemper of
from
asses.
the horses.
Yeah,
from the mules and horses.
Yeah, it's like glanders in horses, right?
Right. It's a similar disease. Okay.
Well, you know what they did? They during the wars, they would take the horses and put the diseased ones, put them on catapults and launch them against the other troops.
Right.
And it's got a pretty low infectious dose, and so it would then take the troops out just by making them ill.
So he is talking about biological warfare way back then.
Yeah.
It's exactly what it is. And that's why it's considered, these are considered to be serious bugs. Anyhow, so Burkholderia grows on agar, does all the right things. It's a regular bacterium in many, many ways. There's nothing difficult about working with it, except, as I say, the pathogenic strains. But the thailandensis species, which is related enough and close enough to the others, is the one that they work here, mainly because it's easier to work with. So first of all, before I get into the paper, I'd like to note that this is work done in various institutions, but the senior author, or the last author, is Jeff Miller at UCLA, and UCLA is blessed with having two Jeff Millers, Jeff F. Miller and Jeff H. Miller. The other Miller, this Miller is a microbial geneticist that's worked on pathogens for a long time, and I know him well because he got this degree at my old department at Tufts. He was an outstanding student, I must say. He's been head of the Department of Molecular Biology and Microbiology at UCLA for some years, and I believe he's going to be an officer of ASM, if I'm not mistaken.
He is president right now.
He's president right now.
Right now it's David Hooper.
No.
David Hooper.
David Hooper, that's right. He's president-elect. I got him.
Yeah.
Anyhow, we're lucky to have him. The other Jeff Miller is the Miller of Miller units. If you do beta-galactosidase assays, you express that in Miller units. That's the other Jeff Miller. Can you imagine can they get their mail straight on one campus, two Jeff Millers? I can imagine the disaster. Anyhow, great people. So here's the story. Burkholderia has a bunch of genes which have been identified, and it has a plethora of the secretory systems named TS1 through 6, which are so fascinating. These are systems where essentially the bug makes a little syringe-like apparatus which can penetrate the membranes of host cells and serves as a passage of toxins or effectors from the bacterium to the host cell to the detriment of the host cell. Okay, so this is they are endowed with that, and so it was thought that one of the main ones, which is called T3SSBAM, you can indulge me in that. There's a lot of names like that. T3 means type 3, and SS is secretory system, so that's not so bad. Anyhow, so there was a lot of question about whether TS33BAM is important for invasion, and it turns out that it's not so easy to study. And the reason is that if you try to determine if it plays a role in later steps, once the bacterium is penetrated, that it does not. So the story starts here, and it's going to go elsewhere. So bear with me because we're gonna move it a little bit around. So how do you study something which is a mutant, which is deleterious in the early steps, but you'd like to find out what it does in the later steps? Well, here's how the photothermal nanoblade comes in. This is a little gadget which allows you to insert a little bubble of liquid inside of a cell without killing the cell. And this is how it works. It has it's a titanium coated capillary. That is, you take a microcapillary, you coat it with titanium, and there's a laser there, and you suck up a little bit of liquid, and when you touch the host cell and you press the button on the laser, that causes a photothermal creation of a little bubble which enters into the cell. It's like a blade, if you want to. I don't know that the word blade is so suggestive, but it's a way of popping a little amount of liquid containing a cargo, and the cargo can easily be a bacterium, into a cell, bypassing, in the process, all the complicated machinery that it takes for cell invasion on the part of the bacterium.
And it's probably more efficient than electroporation, which, you have to treat the cells funny and you have to do all sorts of things. This was really novel.
I don't think you can get a whole cell in by electroporation, can you?
No.
I mean, it's good for getting DNA in, but not the whole cell, not the whole bacterium.
And also electroporation kills about 95% of the cells,
right?
So this, Elio, for this you have to look under the microscope and do individual cells, right?
Yeah, it's one at a time. It's not a high throughput technique, at least not yet.
Someone will figure it out.
Anyhow, so the conclusion of all this hoopla is that this protein, this system, T3-SSBAM, is not involved in the invasion, but is essential for escaping from the primary endosome, which is the term for the vesicle in which the bacteria find themselves upon invasion. That's before they leave the endosome. Leaving the endosome requires proteins which are secreted via this T3-SSBAM apparatus. Okay, that's actually the part of the story that they seem to love and they seem to be totally excited about. And it's probably true because there's been a lot of concern about this type 3 secretory system. But they went on and they found something quite different and they found something quite novel. And the novel thing is that the Burkholderia inside of the cells moves, again, at very high speeds. They clocked it at 20 microns per second. And they, for reasons that I'm not sure about, looked for a mutant in the protein which usually is involved in polymerization of actin at one pole of the bacteria and pushes them around. Okay? So they found that this did not interfere with the virulence of the Burkholderia. So they looked and they found that Burkholderia, in fact, has flagellar motility, which is not surprising. In other words, under the microscope, all by itself, it's going to move just like so many other bacteria move. And that mechanism is due to what was thought to be a cryptic flagellar system, which they identified, called FLA2. And that FLA2 is what makes the bacteria move. And they move. And move they do, as I say, at a high speed.
By the way, Elio, did you know that 20 microns per second is 4 times 10 to the minus 5th miles per hour?
Now I know.
It's really slow, just to give people a perspective here. It sounds fast.
Sure, but in body lengths.
Yeah.
Yeah, but an animal cell is often about 20 microns across, right?
Yeah.
Yeah.
That's fast. Yeah.
So, even though relatively speaking it's moving slowly, it's zipping across that animal cell in no time.
Yep.
Exactly. And in terms of body length, as I say, if the bacteria are about 1 micron, this is 20 body lengths a second. That's not so bad.
Yeah.
And they have a really neat figure. For those of you following along in the paper, if you look at figure 2, They actually cartoon this with incredible confocal microscope images along with the cartoon describing how the nanoblade and the insertion actually worked. So take a look at figure 2 from that PNAS paper and you'll see the incredible descriptions that Elio is describing.
I want to just point out that these are done in eukaryotic cells in culture. They're human embryonic kidney cells. They're 293 cells, which in virology we use all the time for various reasons. But the cool thing that I found here was that when you add these bacteria to the cells, they form plaques on the monolayers. Mm-hmm. And I'm very surprised to see, plaque-forming units per milliliter used to describe a bacterial infection of cells. I'm used to that for viruses, of course, because that's how we quantify viruses, PFU per ml. And these make beautiful round plaques on these monolayers. Because they are killing the cells that they are replicating in, right?
Mm-hmm.
And it goes back to how, when we were describing how the microbes move between the eukaryotic cells, you can begin to see why you're getting a circle or a plaque on the agars because of how they're moving. They're moving outwardly. Whereas a viral plaque will typically stop, where these bacterial plaques may continue to grow as the bacteria continue to move.
Right. Well, let me talk. You brought up appropriately, Vincent, the killing of the cells. So here's the story. The bacteria don't kill the cells very rapidly. They take a while. But they do something very unusual. They cause fusion of adjacent cells. So this is something new, because if the bacteria induce fusion of two adjacent cells, they can go into the next cell without having to go through all the hoopla that the actin-driven bacteria have to go through via the filaments, the protrusions, and so forth. So it's an open space all of a sudden for them. And how this fusion comes about, nobody knows. In fact, this is interesting because fusogenic proteins are of interest to virologists, aren't they?
Absolutely. Yep.
So here is a bacterium which is fusogenic, and it may be the first one. But and also, what happens is that after a while this stops, and when the host cells become large enough, then they pop. Okay, so there's a lot of time and a lot of room for the bacteria to grow. And in fact, in the figures that Michael was talking about in the paper, there's really a lot of bacteria accumulating there. So anyhow, there are two novel things. First of all, I didn't make a point of it. Flagellar motility inside the host cell. I think this is the first case that describes that. Okay, until now, all motility inside of cells was supposed to be driven by actin polymerization. This is news. This is news in itself. Flagellar motility. Who would have thought? Or on the other hand, you can say, why not? But here they are. And the second thing is that the transmittal to adjacent cells is not via poking holes in the next cell, but by causing the adjacent cells to fuse. So that's really a couple of novel things that this guy does, Burkholderia thailandensis, does not follow the book.
This movement by flagellar motion is surprising to me because the cytoplasm is very crowded of a eukaryotic cell. It's not easy to move around. And in fact, viruses move within the cytoplasm on the cytoskeleton because they can't just diffuse. It would take them too long. So the idea that these bacteria are swimming around is a little surprising.
I was thinking the same thing. I was trying to think back to my college days and Reynolds numbers and things like that. It just seems like an unlikely thing to be able to do. I don't know.
Yeah, considering that if we were swimming inside cytoplasm, it would be the equivalent of us swimming in corn syrup.
Yeah,
because of how viscous it is. And when I read this, I was really quite surprised because I always considered the eukaryotic cell insides to be like a no-wake zone for boats. You don't really want to, motor through it because of the wake that would be caused by the microbes moving. And his cartoon alludes to that a little bit. He's showing a wake as the microbes are moving, and it's probably causing some of the collateral damage that is ultimately responsible for, killing the cell.
You wonder if the Burkholderia cell doesn't have to secrete something that is breaking down something at the front end. Maybe. You know what I mean? It seems like they might find something like that eventually.
Well, remember that the physiology of Pseudomonas / Burkholderia is such that it doesn't use anything preformed. It breaks everything down, which is why they're the phenomenal oxidizers that they are. Yeah. They bring in everything and then they make everything from scratch. They truly need to be the pluripotent cell, because even if you give them preformed amino acids, they'll take them, they'll break them down to their basic building blocks and rebuild them from scratch.
So they do have lots of extracellular proteases and things.
Yes.
Like that. Yeah. Okay.
They have elastases, and that's all part of their quorum sensing bit of how they talk to one another. Be interesting to see how Jeff takes this with his colleagues to find out if there are quorum molecules going on inside the cells, and whether or not they play a role in the expression, because you would need much less of the soluble factor than you would if it was extracellular. So there's a whole slew of experiments now that they have this technique worked out that I think we're going to begin to see come out.
Yes.
Well, that's certainly true. The surprises don't cease. The whole idea of the movement inside of a viscous material is really still open for discussion, and bacteria have solved that problem. It's just we don't know how they've done it. Do you recall a paper many, many years ago by a physicist, Purcell at Harvard? It's called Life at Low Reynolds Numbers. It was a fabulous paper. Look it up. It's an old paper, but it's a classic. And he calculated that if the Queen Mary had the same problems that bacteria would have in the same viscous environment, if you turned off the Queen Mary's machinery, suddenly it would advance 2 inches. That's how viscous it is. So bacteria do something that we don't know about. I have to recollect something from my youth. Is that permissible here?
Of course.
When I got
that
drafted,
paper came out in 1973.
The Purcell paper?
Yeah, the Purcell paper.
What's the name of that again?
Life at Low Reynolds Numbers.
Okay. Put that in the show notes.
I got drafted into the Army of the United States. That's many, many, many years ago. And I was sent to Walter Reed Army Institute of Research and told to work on rickettsia and figure out how to divide. And to do that, I was given cells in culture infected with rickettsia. And I would choose what looked like a rickettsia and take a picture every minute. We didn't have a movie camera. It was all done by hand. And I did it until I found that the rickettsia divided into 2. That was nice. It wasn't known at that point. But what was happening is I was looking at the rickettsia. They didn't stay put, but they moved around. They moved around in exactly the same way that the acting movement was then found to be the mechanism thereof. So I had no idea why they moved or how they moved, but I saw them move. And let me tell you, it is a fantastic site. Now, there's a website by Julie Theriot at Stanford. That's Theriot. Juliet, she has incredible pictures of listeria moving, some put to music. I don't know, you really have to go look at it. So let's add that to the list of things.
Okay, will do.
Okay. Well, this is about it. I mean, this is news in various ways. The technique is new. It was not used for one more thing, forgot. The fusion of cells requires the activity of a different secretory system. It's called T6SS, and that was discovered in Vibrio by Makellanos. And so we don't know what it is that's secreted that way, but it turns out to be required. So the pieces of the puzzle are beginning to fall in place.
I have a very nice model for this, which is actually in the supplementary data. It's figure S4. Model of the intracellular life cycle, which is very nice. And I notice at the end of the paper in the discussion, they say this model is based on observations with 293 cells and may not take into account factors that are required for survival and replication in professional phagocytes. So maybe the reviewers made them add that. A little qualification. I want to just point out that this idea of bypassing steps in infection to try and tease out the genes needed for each step is something we do also in virology. So, for example, when viruses infect cells, they bind to receptors. They're taken up by endocytosis, and then they get out of the endosome and replicate. And if you want to dissect the requirements, what you can do, you can put the viral nucleic acid directly into the cell. So you bypass all the entry steps. And it's sort of a similar thing to what they're doing here, except they're putting, of course, the whole bacteria in. But in the case of viruses, we have an easy way of doing it. We can extract the nucleic acid and then insert it into the cell by transfection. So in theory or methodological approach, it's a similar type of experiment. All right. Thank you for that. So, Michael and Margaret, this nanoblade approach, I presume, would be useful mainly for bacteria that replicate inside of cells, right?
Well, I'm also excited about the fact that he says that, it would be useful for molecules as well as bacteria. So, in the squid system, we've been stymied with genetics. And so we have great genetics in the bacterial symbiont, and we've had a hard time with other methods. So maybe something like this. I think it would take... I'm going to wait until you can blast a number of cells at one time.
Yeah, one at a time.
Yeah, you may be able to load up the equivalent of an episomal genetic element into the squid.
Right.
That, the adenovirus equivalent for squid.
Yes.
I mean, there probably is a virus out there that would behave like adenovirus and do transgene expression, so you could ask questions about transgenic, if you overproduce a particular protein, what's the phenotype in the squid?
Right.
But getting that episome out of the ocean, you may be able to just do it with the techniques of our next paper.
All right, our next paper is in Science, July 15th, 2011. It's called Precise Manipulation of Chromosomes in Vivo Enables Genome-Wide Codon Replacement. And this is also a very large team from Yale, MIT, Harvard, and the senior author is George Church, who I know from years ago. Just a little reminiscence. George Church used to work with Walter Gilbert in the early days, and he helped develop DNA sequencing techniques. And now he is moved on to, he still does a lot of sequencing. He actually is heading a project to sequence 10,000 human genomes. And he's developed a lot of technology to do that. So it's not surprising that he develops the technology in this paper, which I would characterize as a massive way to genetically manipulate E. coli. So you want to start this off, Michael?
Yeah, I'm going to take a crack at this. This paper will take you about a week to digest because of the richness of the techniques, and you have to refresh your knowledge of bacterial genetics because they use some of the old tricks from the 70s and 80s that I think many of the molecular biologists of today had forgotten. And so the title is The Precise Manipulation of Chromosomes in Vivo Enables a Genome-Wide Codon Replacement. And the first question I always ask is, why do they do this? And their answer in their paper is they wanted to see if they could. Mm-hmm. And so their hypothesis is, as many of you know, there are 3 stop codons in the genetic code. There's the amber, ochre, and opal. And a little bit of history, the amber codon, which is UAG, or in the DNA TAG, was originally described by Harris Bernstein. And the reason it's called amber, this is your trivial pursuit question for lab. The reason it's called amber is because Bernstein in German is amber. And Harris Bernstein was in a debate with Richard Epstein and Charles Steinberg over how protein synthesis would stop, and the evolution of amber, which is UAG, opal, which is UAA, ochre, which is UAA, and opal, which is UAG. Sid Brenner gave the name amber and ochre to UAG and UAA, and the last one, opal, UGA, or called umber. When I was learning my DNAology, as it were, I heard that story, and a little bit of trivia never hurt you. And the reason they're interested in this is both the amber UAG and opal UAA codons require something called releasing factor 1 in order to stop protein synthesis. We all know that the stop codon tells the ribosome to fall off, and it's really an elegant mechanism, and they need a genetic product, which is the consequence of the production from the PRFA gene, which is releasing factor 1, and that facilitates the stopping from UAA and UAG. The other two codons, or the redundancy in our genetic code of UAA and UGA, the ochre and the opal, require releasing factor 2 to release. So the UAA codon, if you will, is the universal one, because releasing factor 1 and releasing factor 2 will cause it to stop. So they wanted to ask the question, could they replace all the TAG codons within the chromosome with TAA, and then would that eliminate the need for releasing factor 1? So now, the first thing they have to do is they have to go to the genetic code, and the organism of choice that they used was E. coli, because we have a lot of genetics on E. coli. We understand it really well, and most importantly for their technique to work is they have genetic strains which will facilitate their MAGE, M-A-G-E, which is their high throughput replacement scheme, and their CAGE, C-A-G-E, which is their way of facilitating the combinatorial action of moving these mutations. So the first question they asked is how many TAG codons were in E. coli? And the number is 314. Now, I remember making mutants once upon a time in E. coli, and it's hard work. But they have figured out how to really go after this in a multiplexed automated genome engineering approach where they call up their favorite oligo maker and they make the appropriate number of pieces parts. And here's where you have to relearn your genetics and strainology from E. coli, because what they used is they used a strain of E. coli that was deficient in MUT-TS, which facilitates recombination, but it also has the downside of being able to introduce mutations. And they also had in this strain the lambda red gene, which also facilitates recombination. So the way they were able to really facilitate this is by using good strains to do it. Now, we all remember our early days of conjugation when you mated E. coli that had an episome with the fertility factor. It had an ORI-T on it, origin of transfer, and of course the episome was about 2 minutes in length, and then that's how we knew that it took 2 minutes to transfer across, because the only way you got recombination in the recipient is that you would detect a phenotype that was encoded on the episome. But they did a little trick here, and they cheated, and they really made their ORI-T very small, and they also put a selectable marker on it as well, the kanamycin. And when they mated it into the recipient strain, there was a selectable marker on it. So the only thing they would get back is the recombinant. It would kill off the donor, and it would kill off the recipient by the selectable markers that they had. Now, the magic occurs when they have all their codons that they're swapping out with their oligos, and they're building this. And they did some math, and it's very important that you know your math now to know how to truncate things down. And they split the genome into 32 regions, and each region had 10 TAG codons. Now, it was an iterative step that I'm not going to attempt to describe in a podcast. I encourage you to go and look at their cartoons, because otherwise you're going to get pretty lost if you listen to me trying to describe it. But the neat thing is it's an iterative step, and they effectively then invoke their conjugative assembly genome engineering, where they move between 154 kilobases to 2.3 megabases. Now consider that the E. coli chromosome is only about 4.6 megabases in size. So you can see how this conjugative assembly process that they're developing is really pretty remarkable. And the way I tried to wrap my head around this, thinking about how Mother Nature would do this, I equated it to pathogenicity islands. But their true trick is the selectable markers they built into their material that they wish to recombine into the recipient. And they were effectively doing this via what I typically refer to as post-replication repair. They were asking the cell to fix things, and the cell really doesn't like to have single-strand or double-stranded DNA floating around in the cell that's not anchored properly into the chromosome. And so they built into their DNA selectable markers, and they built into their recombinant DNA zeocin, spectinomycin, and gentamicin. And each of those drugs is also pretty clever. Zeocin is in the bleomycin family, which intercalates into DNA. So right there, one of their selectable markers that they're putting into their plates is effectively going to probably facilitate some of this recombination because it's going to mess with the DNA. And so it's actually going to create a lot of single-stranded DNA inside the cell as it, as they have it at a lethal concentration. The other one is spectinomycin, which is in the family of the aminoglycosides, and it binds to the 30S ribosome and interrupts protein synthesis. And then the other one is gentamicin, which is also a bactericidal antibiotic that also interacts with the 30S ribosome, and it effectively causes protein synthesis to stop. And so as they began to slice and dice using these genetic tricks, they were able to begin to replace the codons. But then they bring up a couple of caveats in their description. They talk about how challenging it was, that they were worried about essential genes. And an essential gene is something that cannot be deleted, disrupted, or destroyed. And the TAG codon was found in 43 essential genes out of the 314 genes that TAG was being used. And the second caveat that they introduced is that they were concerned about the overlapping second reading frames. Recall that bacteria, unlike our eukaryotic friends, make polygenic messages. More than one gene is encoded on that single piece of messenger RNA. And so they were concerned about the overlapping second reading frame that the TAG was in. And the net consequence there was, of those 39 genes, they were concerned that they would be underrepresented in the recombinants. So they did a bunch of clonings, and they were able to select out 1,504 clones, which was 47 clones for each of the 32 segments that they were going through. And then they did bread and butter sequencing and asking what was the efficiency of their allelic replacement. So their efficiency for the allelic replacement was 37% plus or minus 19% after 18 cycles. So looking at all of this, you're really amazed at the utility of their system. Now, one of the things that troubled me when I was reading this about substituting out the TAG codon is I know that the TAG codon actually encodes, if you will, for the 21st amino acid, which is selenocysteine. And selenocysteine is encoded by TAG, and it's the selenocysteine immediately downstream of the TAG code is a selenocysteine insertion sequence, which is abbreviated SECIS. And this element is immediately after the UAG, and the selenocysteine insertion sequence happens to be the same genetic element in the archaea as it is in the bacteria. And in E. coli being a facultative anaerobe actually has quite a number of genes that actually require selenocysteine. The best one that I always remember is the formate dehydrogenase, and as any bacteriologist will recall, is that E. coli is a mixed acid fermenter, and it needs fumarate dehydrogenase to do its magic at the end in order to evolve hydrogen gas, which is one of its end products. Some of the other enzymes selenium facilitates is glutathione peroxidase, thioredoxin reductase, and some hydrogenases in E. coli. And selenocysteine is this remarkable amino acid. It has a lower pKa than cysteine. It has a higher reduction potential than cysteine, and E. coli doesn't like to keep it around. It keeps it as hydrogen selenide, and then it
but just... But the point, Michael, the point here is that it uses the same codons as termination codons, right?
Correct.
That's why you're mentioning this.
That's why I'm mentioning it. And so it all goes back to engineering. Is this a good thing? So
how often? So what you're saying is that a fraction of the time the TAG is recognized by a selenocysteine-containing tRNA, right?
Correct.
How often does that happen? Do we have any sense?
Well, it depends on which complement of genes there are in it. They didn't comment on this in their paper, and I didn't have an opportunity to go off and find out how many selenocysteines, how much selenium was used in E. coli. I know about the glycine reductase, the thioredoxin reductase, and the formate dehydrogenase, but I don't know how many of those codons that E. coli needs.
So they ended up changing each of the 314 individual strains, and they said those were phenotypically normal.
Correct.
But then...
Well, almost, or at least almost. They show the growth rates. They're slowed down a little bit.
And then they were able to combine...
In nature, they wouldn't compete. But
yeah, that's absolutely right. Fine. Then they were able to combine 80 changes, 80 TAG to TAA changes. That's the most they were able to get. They never got all 314 in one strain.
No.
And that strain, they said, had some problems. So maybe partially due to getting rid of selenocysteine incorporation.
And they're really doing a lot of chunking of, where they're chunking a big piece of the chromosome in their allelic specific recombination. And they, in the supplemental method section, they actually describe in great detail of how you can actually calculate how much of these things that you have to do. If you don't want to replace all 314 codons, but you really just want to replace a couple, 3 genes, they really give you the primer of how to efficiently do this if you want to over-engineer. And some of their earlier papers, they were actually using it to over-engineer and mass-produce a large number of recombinant proteins.
Right. I mean, I think that's the eventual goal, is to make very different proteins. What struck me here is that they didn't really finish the work. They didn't do all 314. They were only able to change 80. And they decided to publish at this point. Maybe they couldn't get past 80 changes.
I still think it's remarkable. absolutely remarkable. And it reminds me, when I read this, I thought, Craig Venter had done this, sort of artificial system in which, he went about trying to synthesize a bacterial cell, de novo. And here we have whole-scale manipulation of a genome. I think it's just phenomenal to think what this is going to open up, this sort of manipulation.
And that's how they end their paper. They say, and the quote is, in contrast to in vitro genome synthesis,
right,
and or transplantation mechanisms, what they're doing here is they're doing editable and evolvable templates, and they're generating targeted and combinatorial modifications across many, and they estimate around 10 to the 9 genomes in vivo.
Isn't that phenomenal? I just think it's, I can see why they were excited to publish even what they had.
Right. Yeah, the methods are here, and they clearly work. I want to take a stab at describing, just giving an overview of how they did this. So as Michael said, they divided the genome into 32 segments with 10 mutation sites each. Then they made the oligonucleotides to make those changes. They electroporate the oligos into the bacteria and then let the bacterial DNA replication incorporate the mutations into the genome. And they do multiple cycles, and periodically they sequence, or they do PCR to identify bacteria where there are the desired changes, and then they take those and they resubject them to the same process. So they do it over and over again until they get 32 strains with 10 changes in each one. And then, as Michael said, they use conjugation to combine all the changes. But they didn't get all 314. They only got to 80.
Well, E. coli is unique in that it fixes mistakes it perceives. And so while we may want something to happen, E. coli doesn't always do it for you. It'll actually keep what it wants. It's still pretty finicky about what it'll let us do to it in a wholesale situation.
So they said in the beginning they developed this technique because the Venter process, where you basically do whole genome de novo synthesis, it's expensive and it might not be accurate. So here we take a genome template and we use it. But my sense is that this is pretty hard as well, time-consuming and costly. I don't know. What do you think? Do you think? I mean, just let's say the conjugations. How long does it take to do a conjugation?
It really depends on it. In principle, you need the right background strain in which to mate it, and you need both the positive and negative selection markers that you want to take out. They happen to use the tall C, which they can counter-select with colicin, and so I think that's what they were going. They had the right strains in their armamentarium to go into a genetic system where we don't have as much genetics like E. coli. It's going to be much more challenging for them.
I think
aside from Bacillus, which has a fairly well sophisticated set of strains out there, I think it's going to be very challenging. Where I think the exciting parts can come is if you go to Streptomyces and the antibiotic producers. Mm-hmm. Because if you can begin to tinker with the antibiotic producers, we may be able to do variations on the theme on making new antibiotics or tinkering with some of the precursors to help the big pharma make new drugs.
I view this as space travel, right? Not everybody's going to do it, but the technology is going to be used by a lot of people eventually.
Very good.
Elio, what were you going to say?
Well, I was going to say this could be called biological combinatorial library. Creating a combinatorial library in vivo instead of by organic chemists and
Exactly.
Yeah. But listen, I want to congratulate Michael for this masterful exposition. I'm glad there isn't a quiz at the end.
Oh, God help me.
I agree, Elio.
This was... Fortunately, I was on vacation this week, so I could actually spend some time on this.
It's amazing. It's a technical tour de force.
Explaining it as clearly as Michael has done does us all a lot of justice. So
I agree. I mean, that was just phenomenal.
But what we need to remember is, you know, to the listeners out there, in the back of every molecular biology catalog, there's a list of strains. And if you're bored at lunch one day, take a look in the back of your favorite restriction enzyme company and look at the strains that you can actually purchase. I mean, a lot of them will have some of these mutees in them, and actually investigate what some of these genes actually do, because this is the only reason why this paper was possible, is because they knew the genetics and they knew how to cheat the system. If you put this into wild type, all bets are off, because E. coli is not going to recombine at the frequency that they were getting it to go at. And the lambda red, you know, one of the things I forgot to point out was they found a landing site in each of their strains. And this landing site was, they called it the safe insertion. They had another letter after it, but I don't remember. It was a safe insertion site, which means that there were no coding genes or regulatory elements within it. So this really requires you to have your genome not only completely sequenced that you want to tinker with, but it's got to be completely annotated. That means you have to understand what each and every part is doing. And so, you know, we say this almost every week. It's a great time to be a microbiologist, but in reality, the technology that these folks have just laid out before us will really give us an opportunity to ask some really interesting questions about microbes like Margaret's working on. I mean, can you imagine if you were able to do this with your Vibrio?
Oh, that'd be great.
And tinker with the quorum sensing parts?
Oh, yes, absolutely.
Do you have a genome sequence of Vibrio fischeri?
Yeah, actually we have several strains are sequenced.
Good. Well, you better start working on this, Margaret.
I know, I got to get right on.
But this paper goes back to how we began to regulate biology in the 70s. This was what everybody was concerned with. And this crosses into the dark realm of dual use. And this technology really opens up the potential that people who don't have the best intentions can, you know, take strains and manipulate them to their own purposes. But the good news is you have to really know what you're doing in order to get this system to work as they clearly laid out. And it's not as trivial as, you know, you may think it's easy in principle, but the bacteria are very clever and they make it a little bit more challenging.
Well, also, as we see here, you make what you think are going to be innocuous changes in the fitness declines of the bacteria. So
that's right. That's the point that's important. Even though the growth rate is close to normal in this construct, it's not. And this matters. In nature, it would matter a great deal.
Yeah.
Well, it goes back to that famous Small Things Considered post by Elio's good friend on Luria Bertini broth.
Yep.
Oh, yeah.
You really have to, if you haven't read that one in a while, go back and take a look at Bertini. Take a look at it, because
it was written by Hiroshi Nikaido, who should know.
That's right, and it's you know, really tells you some wonderful things and how lousy LB broth is, which I was really surprised why they used LB broth, but you know.
And this
that's what geneticists and molecular biologists do. A little bit of cell physiology would go a long way in some quarters, but that's asking a lot. Anyhow, I think this was great, really.
Okay, thank you, Elio. Thank you, Michael. Let's read a few emails before we finish. The first one is from Mary Ewell, your friend there, Elio, right?
Yeah, my pal.
She writes, hi, I've been listening to the evolution of TWiM week by week, and I want to say that I am delighted by the direction it has gone. I listened to number 11 yesterday and found the format especially informative and interesting. By format, I am referring to the selection of, in this case, 2 papers, both of which were introduced well to orient the listener to the context and the topic. In each case, there followed a real discussion among the co-hosts that was accessible to listeners with varied backgrounds. Acronyms and specialized terms were defined or replaced by more common words. As if that weren't enough, the viewpoints presented this week were particularly relevant and important vis-a-vis our microbiota, allergies, antibiotic usage, agricultural practices, and such. This information needs to be widely disseminated, and TWiM makes an excellent contribution to that process. Carry on.
Wow.
Isn't she nice?
It's very nice. Thank you. Yeah, we are evolving. We are finding our center point, and it will take a bit more time. But tell us, other listeners, tell us what you think, because we've done a variety of things. We've done the papers. We've had some more general discussions where papers aren't involved. Tell us what you like, maybe a mix. You can send us your email to twim@twiv.tv. The next email is from Jenny. I'm Jenny, an RN who has very much enjoyed your work. Or is this play? It's for TWiV, TWIP, and now TWiM. I don't know, Jenny. That's a good question. It's both, right? We are very fortunate to have this kind of work that's a lot of fun. Thanks for making fascinating subject matter approachable and fun. I've listened to your programs while traveling and caring for ill and dying family members. Your voices helped me use the solidity of science as a kind of ballast during difficult times. That's very nice. You'll probably pick and choose from this email what to discuss, since I know I'm asking too many questions. Enjoy the choice. Well, I'll read them all because they're good. Going back to TWiM number 5, perturbation of Arctic soil using nanoparticles gives me the willies. One of your guests mentioned a thought, spraying human skin with nanoparticles to stop MRSA. Yikes, another shiver of concern. That wasn't me. Was that you, Michael?
It wasn't me. I wasn't on. I think that was Ron Atlas, because didn't you have Ron on for...
I think he was on that one. Maybe it was his idea.
I don't remember the silver or the nanoparticles with anything.
Yeah, I think would be a bad idea. Probably indiscriminately alter your microbiome on your skin. All right, but wait, she writes, way before I had a foundation for pro and prebiotics, I quietly theorized that skin infections were an imbalance in the living systems that are usually on our skin, or that imbalancing the microbes there may be a prerequisite to skin infection. Also, I understand that when some pathogens invade the gut, one of the first tasks is to launch an attack on the commensal microbes that hold territory there. Back to the skin and nasal mucosa. I understand now that the Human Microbiome Project has identified many, many bacterial species on the skin that live in a complex interdependence on us. I believe a current CA-MRSA therapy is judicious use of antimicrobials and topical intranasal mupirocin, and that limiting use of mupirocin to avoid drug resistance is a concern for this treatment, as it is for all antibiotic treatment. So let me make my case for this idea I have for a study and perhaps personalized medicine. Attempted transplant of a cocktail of known non-resistant commensal skin organisms to the skin and nasal mucosa of a person with CA-MRSA. What's the CA stand for, guys? Community acquired. Thank you. Perhaps after a reduction in bacterial numbers using a method that does not linger, so that the reduction method wouldn't interfere with the colonization of the new commensals. Perhaps drying, perhaps phage. Of course, not being a microbiologist, I don't understand the niche selection pressures for microbes, nor do I get why some colonize and why some are transient.
Neither do the microbiologists. The niche selection pressures are unknown to us, and that's what the microbiome is all about. We're really trying to understand the trigger points, and we started with the simple story of Margaret's bobtail squid and the Vibrio, because I think that's the simplest solution, where you have one organism and one behavior. The skin of a person is extremely dynamic. And another trivial pursuit question for you: the average human drops 2 million skin cells an hour where they sit. So we've been sitting here now for about an hour, so we've left a pile of about 2 million or so where we've been sitting, and each and every one of those skin cells is covered with the microbiome of that individual. And so this is how it moves about, especially like places like hospitals where you're wearing those delightfully stylish gowns where you're dropping probably more than 2 million skin cells an hour.
All right, let's go on here. Related questions. I understand that in some cases, simply by going for a while without anti-infectives, some people clear MRSA because of the lack of selective pressure for resistance, but it's not at all a sure thing. Wonder what the odds are on that clearance and what the factors are. Wonder how important it is that we have resistant microbes in our gut. Is this a super complicated world of many kinds of plasmids, and are those plasmids in communication with our skin and nasal mucosa organisms?
Plasmids move, and
say yes.
Say yes. Do you think they move from your gut to your skin and
Oh,
nose?
yes.
Yeah.
Why not?
Yeah,
Why not?
they do. There have been a number of studies out there that have demonstrated that.
All right, so now she has a few questions on triclosan and household disinfection, which I think we should leave for another episode because
I think so.
we need to get someone who can wax poetic on that. So let's end up with her last paragraph. I leave you with a crazy idea. Of course, I teach people to wash hands just before leaving work. How about washing, carefully drying, then recolonizing them with a complex normal flora on their way out the door, kind of like a handshake? I can imagine one problem with this crazy idea. The complexity of microbes can't live off the human body long enough. I suppose there's nothing like a human handshake to transplant human commensals. Alternatively, after washing and drying, apply a solution that selects not so much against MRSA, but provides conditions that favor commensals, kind of like a prebiotic for the skin. We would want to know what conditions make it a little shaky for pathogens while providing the best chance for beneficial commensal recolonization. Then that recolonization might happen next time you touch another person, like your child when you come home.
It reminds me of somebody, I believe in Australia, who claims that the skin bacteria are so unique and so different that they could be used forensically instead of fingerprints. So if that's true, the situation is very complicated.
Yeah.
In fact, you can do that with staph epidermidis. The staph epi is almost unique to the individual. The bulk staph epi on the average human. When they began to do the MLSE typing of it, they found it was much more varied.
Is that on all body sites?
I'm trying to.
You know how Marty Blazer and those guys, when they were doing the arm, they saw some around some specific communities at the wrist and some specific communities as you moved up the arm and the axle of the arm and so on. Is that particular microorganism specific to one site?
I'm trying to remember where he was doing this. I remember going over sequencing gels. This is the my brain is just fried. I'm trying to remember his name. He's a very famous Japanese worker who sequenced the first Staph aureus. Hiroshi, what is it?
It's not Umasaki, is it?
No.
He does gut microbiota.
He's a symphony conductor for
Ozawa? Seiji Ozawa?
Not quite.
No. Not quite. I can see his face.
How long ago was it done?
This was done about 2007. He was tinkering with it in 2007. I saw the raw data.
Keiichi Hiramatsu.
That's it.
That's genome sequence of Staph aureus.
Yeah.
This is 2008, along with Olaf Schneewin.
Yeah, that's it.
All right, there you go. Keiichi Hiramatsu.
That's it.
I like saying Japanese names almost as much as I like saying Italian names. Jenny says, thanks for listening to my silly questions. They're not. They're good questions. And we are glad you're thinking of this stuff. Jenny is from Oneonta, New York. All right, that will do it for another TWiM. You can find us on iTunes at the Zune Marketplace or at microbeworld.org. We also have an app for your iPhone or Android device that you can use to listen to the episodes. You can find that app at microbeworld.org/app. If you do go to iTunes and listen to us there, leave a comment if you haven't done so already. You can just say you like TWiM or whatever you feel about it. It helps us to stay on the front page of the Medicine Podcast Directory. The more comments there are, the more likely it is that we stay on the front page. As I said before, we love to get your questions and comments. You can send them to twim@twiv.tv, or you can go over to microbeworld.org/twim and leave a comment there. Michael Schmidt is at the Medical University of South Carolina. Welcome back from vacation, and thank you for being with us today.
Well, thank you, Vincent. It's always fun.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
A pleasure.
And Margaret McFall-Ngai is at the University of Wisconsin. Thank you, Margaret.
Oh, you're welcome. And thank you, Michael and Elio, for those great reviews of these papers.
It's easy when you have good material.
It's true. It's good stuff. I'm Vincent Racaniello, and I am at virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM Communications, Director Barbara Hyde, Chris Kondayan, and Ray Ortega for behind-the-scenes help. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.
Michael gets a special praise this time.
Thank you. That's high praise coming from you.
You do go on, but you go on very well.
I go on very well.
That's a good one. I like that. You do go on, but you go on very well. That's great. Thanks, everybody.